Welcome to the NIIMBL American Rescue Plan webinar series. This webinar series highlights the projects funded through the American Rescue Plan. The goal of these projects is to help the nation prevent, prepare for, and respond to coronavirus public health threats. In this webinar, we will hear from Eric Munson from Purdue University on the project Advanced Characterization and Manufacturing Methods for mRNA Vaccine Development. You can find additional webinars in this series on our website, www.niimbl.org. And now we will turn it over to Eric.
Thank you for the kind introduction. So let me go ahead and tell you a little bit about our project and begin by starting with the team. So the team of researchers that we have are at Purdue University. They include myself as the principal investigator for the project, and Dr. Elena Alexenko, Dr. Elizabeth Topp, and Dr. Tony Zhao. And so each one of them contributed in a very unique way to this project to be able to enable us to successfully accomplish what we did. So what did we work on? So one of the challenges that we face with mRNA vaccines is the fact that they typically have to be shipped at very low temperatures. So many of you have heard about the ultra cold freezers shipping at minus 80 degrees Celsius, and it's a significant problem. One of the issues can happen is that if the vaccines warm up, we don't know if they're going to be valid or not, if they will actually be effective to be able to treat the coronavirus. And so one of the things that can happen during shipment is they warm up. We don't know if they're viable. And so there was a specific example when Pfizer was shipping out their vaccines. And what happened is that they could actually monitor a change in temperature while the shipment was being sent over the Pacific Ocean. And they notified people of this and said that they're going to go ahead and just send another shipment because they couldn't be sure that, in fact, the vaccine was going to be viable, even though it only warmed up to, say, by a few degrees from minus 80. So what we'd really like to do is to solve this by not having to have this particular cold chain. So this is a slide that really emphasizes the challenges that exist with cold chains and why we go through this pretty exhaustive formulation process for things such as mRNA vaccines. So generally speaking, if a pharmaceutical company is interested in coming up with a particular drug substance, what they'll do in formulating it, what they'll do is they'll start with a frozen formulation. The advantage is that they can do this very rapidly, and it's going to be relatively stable as long as you keep it very cold. The downside is that you have this extreme cold chain like you had with the mRNA vaccines. It's really not an optimized formulation. It can potentially degrade like I just described. And so the next step beyond that is to create a formulation that is in the solid state. In the solid state, or what we call the lyophilized state, it takes a little bit longer to develop, say from years 2 to 5 once you come up with a formulation. The advantage is that it's very stable and it's less impacted by the cold chain that you have. The downside is that it can be expensive and time-consuming process and is also somewhat low throughput. Ideally, what you'd like is a liquid formulation, but of course we couldn't have a liquid formulation because we couldn't wait 5 years for the coronavirus to be stabilized. We need to have a formulation right now, which is why they came up with the frozen formulations. What we'd like to do is to at least get to the point of having a lyophilized formulation in the future to address the ability to get the vaccine out very, very quickly, but also stable. So two areas of research that we worked on were, one is called freeze drying or lyophilization. That's a very reliable method for producing stable formulations in the solid state. The downside of it is that it's relatively low throughput, but it's very, very well accepted in the industry. One of the downsides is that we couldn't actually come up with enough lyophilization capability or freeze drying capability to come up with enough doses for people to have doses in a reasonable amount of time for the mRNA vaccines. And so because of that, we also looked into some alternative strategies such as spray drying, which has a faster throughput, but making sure that remains sterile, which is one of the things that we have to do, potentially can be an issue with spray drying. We actually investigated both. We found that spray drying was not really a good alternative. compared to freeze drying. And so basically we went with the freeze drying approach to try to understand the stability. So the first thing we did is we tried to see, is there a formulation that is stable in the solid state? And so what we did is we tried, we basically made formulations using standard industrial practices, and then we added different amounts of sucrose, which is a very common pharmaceutical stabilizer for these systems. Then we did the freeze drying or the lyophilization, and then we compared the stability that existed either at 4 degrees Celsius, which is essentially a refrigerator, and then just leaving them at room temperature. So the first data that we got was on the 4 degrees Celsius data. And not to get into a lot of the details here, but generally speaking, you want to have a high number here for what we call the encapsulation efficiency. And the solution formulations, which are the solid bars here, you can see that they function quite well. In fact, they go all the way out to 6 months, and they have very good encapsulation efficiency. Interestingly, the lyophilized formulations are not quite as good with respect to their encapsulation efficiency, but at least in the right ratios, they remain relatively stable also over a 6-month period of time. The other thing that we try to do is to make sure that these nanoparticles, so in other words, the virus is put, or the vaccine is put into these nanoparticles, and these nanoparticles have to remain just the right size to be able to get into a person's cell. And you can see that we actually managed to do that as well for the solution formulations. They work pretty well, except for a formulation that, except for the sucrose sample up here. But generally speaking, they maintain the particle size, which is good. The lyophilized or the freeze-dried formulations also work pretty well as well. And so this is definitely promising. In fact, with a lot of these mRNA vaccines now, they do say that they are stable under refrigerator conditions for several months. And so once again, this data also reinforces that. Now, if we want to think about doing that for a freeze-dried sample where we'd like to potentially store it at room temperature, you can see that, for example, the solution formulations, they did pretty good up to about 3 months, but after 3 months, they really were not stable anymore. And you can see that because the encapsulation efficiency went way down. And then you can also see that essentially the particle size went way up for almost all of the formulations. And so the net result is that with a high particle size and the fact that encapsulation efficiency went down, the issue associated with solution formulations is that at room temperature, they just wouldn't be viable for that 6-month period of time that we would like. Now, that being said, for the freeze-dried formulations, you can see that really the encapsulation efficiency didn't really change. So they were actually quite good for these 5 and 10% sucrose samples. And you can see also that basically the particle size didn't change that much for these formulations either. So in fact, what we discovered is that you can make a room temperature stable mRNA vaccine formulation by lyophilization that is going to be better than the solution formulations. And so we're really quite excited about that. So in addition to just understanding some of the basic stability of these systems, we also went to sort of the next level to try to understand, can we learn more about how the mRNA vaccines function? And specifically here, we're using a technique called solid-state NMR spectroscopy, but not to get into the details. What we're really interested in doing is understanding, we've got systems where we've got the vaccine, or in our case, we've got yeast RNA inside of one of these LNP nanoparticles. And we're trying to understand, when we create these in the solid state, do we know is the mRNA on the outside or on the inside of the particles? And the bottom line is that we were able to, by using a combination of NMR spectroscopy, tell the difference between samples that were basically containing mRNA on the outside versus mRNA on the inside. And you can see here, it's really highlighted here by this blue section here. The NMR spectrum is very different for a sample where the mRNA is not inside of it, as opposed to all these other ones that contain either no mRNA or different loadings of mRNA. We can definitely tell the difference between the two. And this is quite important for people who are trying to make these mRNA LNP formulations to know when they put the mRNA into a lipid nanoparticle, is it going to be stable or is it not going to be stable? So some conclusions that we have from this research include that we were able to look at creating room temperature stable formulations. using lyophilization. We found basically that the 5 and the 10% weight per volume sucrose samples were better than those for the solution NMR. And so the idea is that for the next sort of level of the pandemic, if we need to have samples where they can't be stored under a cold chain and we had to send them out at room temperature, could we do this? And the answer is yes, potentially we can, based upon the research that was shown here. And so this will really help people, companies that are developing sort of the next generation of mRNA LNP vaccines, where they need to get something out very, very quickly. Can they do that in the lyophilized state? And the answer is yes, they can create these vaccines that will function in the solid state. With that, I'd be happy to answer any questions that may exist.
Thank you, Eric, for a great presentation. I'm curious if anything surprised you or anything jumped out as you were conducting this research for this project.
Sure. So that's a great question. One of the things that we found that was really kind of surprising was the fact that these samples were stable over room temperature. And so the net result is that because they were stable at 4 degrees Celsius, you could actually store these vaccines and have them be stable for 6 months in a refrigerator. And of course, when we were trying to get the vaccines out initially, the pharmaceutical companies didn't have that particular piece of information. And so now that we know that we can store these at 4 degrees Celsius, that's actually quite powerful. There's still a cold chain, but it's not nearly as excessive as keeping them at minus 80 degrees Celsius.
Excellent. Well, thank you, Eric, for the great presentation, and thank you for watching our American Rescue Plan webinar series. To learn more, visit www.niimble.org.